Enzymes are globular proteins. Therefore, there are going to be a number of factors that are going to affect how this protein is going to work. Namely, we're going to be talking about the factors affecting the rate of enzyme activity. I'll list down the factors simply. The first thing is going to be temperature, of course. Then you have pH. Then, of course, the enzyme concentration is going to affect that reaction. The substrate concentration is going to affect that reaction. Now, what exactly am I doing? Placing E and S with these square brackets. How does that translate? Whenever you have square brackets in chemistry, in science, in biology, that means concentration. Concentration. That's the C. Okay. And at the same time, there's something known as an inhibitor. We will talk about inhibitors in the later spread of this chapter, but inhibitor concentration is also a factor that is going to affect how an enzyme functions. So factors affecting enzyme action. Number one, we're going to be talking about temperature. Number two, we'll be talking about pH, enzyme, substrate, and inhibitor concentration. Starting things off, we have, let me reduce the pen size. The first thing that we'll be talking about would be temperature. How exactly does temperature affect the rate of enzyme activity? We're gonna utilize a little bit of physics here as well. We're gonna be talking about something known as kinetic energy. The first statement I will give to you under this would be that higher the temperature, higher the Ke. What does Ke stand for? That would be kinetic energy. Kinetic energy. 3 dots in a triangular form, that means therefore. So therefore, the molecules move faster. Higher kinetic energy. Therefore, the molecules move faster and the rate of collisions, they're moving faster, so they're going to collide at a higher rate. Rate of collisions increase as well. I'll highlight the key points. Higher kinetic energy, collisions increase as well. My handwriting seems okay. I'm gonna have to improve my handwriting right here, right now. Sorry about that. That's the main thing that you need to put into your mind when it comes to temperature. By increasing the kinetic energy, more movement, more collisions. More collisions. There's something else that's going to play into this as well. Now, when they collide, now they have a lot of energy in high temperature. When they collide with residual energy, enough residual energy. What do you mean by residual energy? Extra energy. Temperature is high, they have a lot of energy. So when they collide, they have more than enough energy, which further promotes bond formation. Bond formation, like so. More bonds, more products are going to form. They have enough energy. That's the idea. Now we need activation energy. The reaction to take place when they collide with a lot of energy, what's going to happen is, obviously it's going to go above the activation energy. We have enough residual energy. Therefore, the reaction will go to the product size. Product side. Okay. Is it all on the good side when it comes to temperature? The more we increase the temperature, the better results we're going to get. Is that the case when it comes to temperature and enzymes? Absolutely not. That's not the case when it comes to enzymes. If you go too much, just like with anything, too much is not good. If, however, your temperature exceeds too much, it goes too much, then the hydrogen and all these bonds, hydrogen bonds, ionic bonds, protein bonds. We talked about those protein bonds in the previous chapter. Now these bonds holding the shape break. That does not look like break. Break. And we say that the enzyme denatures. Denatures. I'm going to take a small moment to talk about this. So this is the process known as denaturation. Very simply put, if I have an enzyme that looks like this is the substrate, no, that can perfectly fit into that particular enzyme's mouth, active site, that's the active site, and catalyze the reaction. The moment an enzyme denatures, this is what is going to happen. The active site of the enzyme will change. Now it's denatured. It doesn't retain its shape like it used to. So this thing, it's not a complementary fit. It's not an induced fit. Substrate molecule and the enzyme, they are not complementary to each other. They can't bind. That's the problem. That's the idea of denaturation. Protein denaturation, or more specifically in this case, this is going to be enzyme denaturation. That is how temperature is going to affect all of those reactions. Talk about pH. We all know that pH is a measure of the hydrogen ions in a solution. Simply we're talking about the H plus concentration in any solution with pH. Now these H plus ions interfere with the R groups. We know the side chains, these R groups of amino acids. Each and every amino acid is going to have an R group. If you have too much hydrogen ions, they're going to interfere with the R groups of these amino acids. In turn, that's going to affect the ionic bonding and the 3D structure. The 3D structure. In the same sense, we're going to be affecting the active site. The active site is going to differ. Third factor, E square bracket. I'll lay it out for you here. We're talking about the enzyme concentration. Enzyme concentration. In a perfect world, if you have more enzymes, more catalysis can offer. Higher the concentration of enzymes, if we have, nope, provided, if we have enough substrate molecules, we will have a higher rate of reaction. Higher the rate of reaction. More enzymes, higher rate of reaction. That's the idea. Linearly increasing, but we also obviously have to have enough substrates to be catalyzed. Now, this is the exact same idea with the substrate concentration as well. So, S square bracket. Same idea. Rate of reaction linearly increases. Linearly increases with increasing substrate concentration. DNA increasing substrate concentration. But there's something like this. This statement you're not going to understand yet. I want you to hold on to it. We'll talk about that statement in a bit. But at a certain threshold known as the Vmax. Vmax stands for maximum velocity. We will talk about that in detail. But at this point known as Vmax, all of the enzymes are working at the maximum rate. At the maximum rate. All active sites are filled up. All those active sites are filled up. We'll talk about Vmax in detail. Just hold on to that point. Following substrate concentration, we're going to be having the inhibitor concentration as well. I bracket inhibitor. Just to give you a basic idea of what inhibitors are, just as the name suggests, inhibitors are going to inhibit enzyme activity. That's all. We will talk about inhibitors and inhibition in more detail as well. Until then, that is all you will have about inhibitors. They inhibit enzyme action. What about inhibitors? Now, inhibitors reduce the rate of reaction. Reaction. Reduce the rate of reaction. Obvious, right? If it's going to stop the enzyme, it's going to reduce the rate of reaction. Simply speaking, it inhibits the enzyme. Inhibits the enzyme. Okay, that statement I wrote too fast and you're not going to understand it. Here we're going to be talking about reversible, competitive and something known as non-competitive inhibition. All of that will be talked about below. All of those things will be talked about below. We're going to be talking about reversible inhibition, competitive inhibition and non-competitive inhibition and how all of these things are going to play an effect. Let me pick a new color that we did not use previously. I think a very bright green is going to do. Okay. First of all, we will focus on competitive inhibition. Competitive inhibition. I will type out the bullet points when I come to this. Here's the deal. Have a similar shape to the substrate. Therefore, compete with the substrate for the active site. My God. For the active site. Okay. I am a little rusty, I will admit that. We get back into all of this. Right. Here's the deal. If we have an enzyme that has an active site related to that shape, this is going to be the substrate. No, it's going to be complementary. It's going to fit onto, it will fit onto the particular active site you will find on this. On the same end, we have the, I'll name the substrate S. On the same end, we have something that looks exactly similar to that, but it's not actually the substrate. This is the inhibitor. Now, do you see? This guy can go and bind to this. If that happens, he is not going to be able to bind and form the product. Problem. That's the theory of competitive inhibition. The substrate competes with the inhibitor for the active site. If there are more substrates, they win. They win the war. If there are more inhibitors, they win the war. That's the idea. Mm-hmm. The second point when it comes to this that I have to mention is now that you have the idea, the rate of reaction can be controlled by either the increment or decrement in, nope, not decrement in substrate and inhibitor concentration. How do I spell it? Inhibitor concentration. Meaning, if you have more inhibitors, reaction will stop, reaction will be inhibited. If you have more substrates, reaction will go ahead and you will have more products. What about non-competitive inhibition then? Non-competitive inhibition. This has nothing to do with the active site. They bind to elsewhere, not the active site. Not at all. Not the active site. And then change the shape of the active site entirely. Previously, did we have any form of active site shape change? No. Enzyme was fine. Enzyme was completely alright. Nothing happened to the active site. It was the competition between the substrate and the inhibitor for the active side that led to either the products being formed more or the reaction being inhibited. But in this case, that's not what happens. Here's the enzyme. Here's the active site. And of course, here's the substrate again. But in this enzyme, not just enzyme, in any enzyme, we have another site. This is known as the allosteric site. Just for clarification, this is the active site. That is the allosteric site. Now, in this case, the inhibitor looks something like this. The inhibitor can go and bind to the allosteric side of the enzyme, and then it's going to change the shape of the active site. Then it's going to change the shape of the active site. That's the idea of non-competitive inhibition. Just like the name suggests, usually there's no competition. Substrate is fine. But the thing is, it's going to change the shape of the active site by the binding of this inhibitor molecule onto the allosteric side of the allosteric side. Did I say side? My lord. I think I'm too rusty. Allosteric site of the enzyme. Like so. If I were to draw a small graph that looks something like this. I won't label any axis too much either. So this will just be the substrate concentration, and this will be the rate of reaction. If this is my normal reaction, there's a nice rate increasing and then plateauing, not an issue. If I had a competitive inhibitor, it would look like this. Rate of reaction drops. If I have a non-competitive inhibitor, that is going to be even lower than that. Sometimes you will be given graphs like this to pick out the normal reaction. They will most of the time label the normal reaction. Then you have to differentiate between what is competitive and what is non-competitive. Every single time the higher line will be competitive, lower line will be non-competitive inhibition. Non-competitive inhibition. Now, since we're on the topic of inhibitors and inhibitor concentration, since we talked about competitive and non-competitive inhibition, I would also like to talk about a certain concept that's going to come under inhibitors. This phenomenon is known as end product inhibition. This is known as end product inhibition. If I were to lay this out, the end product. Now we know whenever a chemical reaction goes on, we're going to be getting some form of product. No? So the end product of a continuous chain of reactions bind as a non-competitive, I will explain what this is all talking about, non-competitive reversible inhibitor to control a certain reaction. Okay, interesting. Let's see what that chain means. If I have the substrate that looks something like this, substrate, that's going to turn into product 1. And then later on, this product 1 is being converted onto product 2. Now, in this chain, this is the final product, so we typically call this the end product of the continuous chain. This is the continuous chain, that is our end product. That is our end product. Now, to convert this substrate to product 1, let's say we want enzyme 1. To convert product 1 to product 2, let's presume we need enzyme 2, like so. Now, this is what is going to happen. This end product is going to act as a non-competitive inhibitor onto enzyme 1. Non-competitive inhibition. Meaning it's going to go and bind to the allosteric site. Nothing is going to happen to the active site, meaning there's no competition between the active sites. The active site will change as a result of non-competitive inhibition. Don't mix that up. That's the idea of end product inhibition, and those will be the factors that are going to affect how a certain enzyme is going to work.